Transcriptome sequencing and whole genome expression profiling of hexaploid sweet potato under short stress. Purple flesh sweet potato is one of the most important crops contribute to solve the malnutrition problem, especially in developing countries. Due to soil salinity problem, these plants are unable to cultivate and lead to decrease in crop yield around the world. Transcriptome sequencing technique offering a rapid approach for crop improvement with promising agronomic traits and stress adaptability. Using it, a high-quality, yearly matured cultivar with green leaf and purple flesh-tolerant roots and salt-tolerant is grown in Hochland nutrient. Seedlings were exposed to salt stress by adding 200 micromolar NaCl into Hochland solution for 0, 1, 6, 12, 48 hours. 0.1 gram sample was taken from the upper third leaf after 0, 1, 6, 12, 48 hours from salt-treated and untreated plant for each next and third generation sequencing. Total RNA was extracted from collected sample with the trizone method according to the manufacturer protocol, using new generation sequencing and third generation sequencing to sequence the mRNA. Library preparation for transcriptome analysis. Sequencing libraries were generated using NEB-NEXT Ultra RNA Library Prep Kit for Illumina. Clustering and sequencing. The clustering of the index coded sample was performed on a seaboard cluster generation system using TrueSeq PE Cluster Kit V3 Seaboard HS Illumina. By using second and third generation technology, Illumina sequencing generated 170 million clean, high-quality long reads that were assembled into 15,998 unique genes with an average length 2,178 base pair, and 96.55% of these unique genes were functionally annotated in the NR protein database. A number of 537 unique genes failed to hit any homologous, which may be considered as novel gene. Unique gene S02 and 1120, which are significantly upregulated during 6, 12, and 48 hours of short stress. These two unique genes were aligned to GID1 like gibberellin receptor. Furthermore, number of 4, 479, 281, and 508 unique genes were upregulated with significant expression level in short stress treated samples at the different time point of short stress, including 1, 6, 12, 48 hours respectively. The highest numbers of DEG was induced at 48 hours of short stress. A total of 24,559 SSR and total of 562,174 SNMP between transcriptome were identified. Protein kinase include many genes which are found to play a vital role in phosphorylation process and act as a signal transducer or receptor proteins in membranes. Salt stress tolerant in hexaploid sweet potato plants may be mainly affected by transcription factor, protein kinase, protein detox, and hormones-related genes which contribute to enhanced salt tolerance. Sweet potato is a hexaploid heterogeneous non-model crop with a complex genome lacking high-quality reference genome. High-throughput RNA sequencing is required for identification of candidate genes involved in salt stress tolerance. The regulatory mechanism and salt-resistant related genes in sweet potato. Most of stress-induced transcriptional changes occurs at least after 3 hours of salt stress exposure. Photosynthesis-related gene. Photosynthesis is the most significant physiological process for the plant life and during all growth stages is affected by stress factors. This transcriptome sequencing data of hexaploid sweet potato under short stress condition can provide a valuable resource for sweet potato breeding research and focus on novel insight into hexaploid sweet potato response in short stress. In addition, it offers new candidate genes or markers that can be used as a guide to the future studies attempting to breed salt-tolerant sweet potato cultivars.